So in the last video, we talked about we left off on talking about the different types of RNA. We're going to go a little bit further in. So here we have our mRNA, our messenger RNA, our rRNA, and our tRNA, or transfer RNA. So let's look at each of these individually. mRNA is the actual copy of DNA. This is made in the nucleus and then brings that instruction from the nucleus out to the ribosome, which is out in the cytoplasm. It is a single strand, and instead of the thymines, it has the uracil. rRNA is found in ribosomes. Ribosomes is basically made out of a protein and RNA together. tRNA is the transfer RNA. This is specified by the mRNA. On the mRNA, there are bases. So we have 3 bases here on the mRNA. These match up with the bases on the tRNA. tRNA is carrying an amino acid, which is going to be put together to make that protein. So let's take a look at how each of these is used. Transcription. Transcription is the act of making mRNA from our DNA strand. Very similar to that of DNA replication is how we make our mRNA. We use RNA polymerase as the enzyme to do most of this work. So RNA polymerase is going to open up the DNA, and it's going to put the complementary bases of RNA onto the template strand to make that strand. Since RNA is only single-stranded, we only use one side of the DNA in order to make our RNA. So RNA polymerase will unravel the DNA, open it up. It does look for a specific sequence of DNA. This is called the promoter. This is basically the area that it starts. It copies the DNA into the mRNA. mRNA then leaves the nucleus, and then it needs to be edited before it leaves before it's actually used. So here's a picture of the process. DNA is being unwound. So here is our DNA being unwound. RNA polymerase here is being used to bring in complementary bases of RNA, and our RNA strand is being made with just one side. Remember that RNA has uracil, DNA has thymine. So, like I said earlier, we need to edit our RNA. So we break it up into small little pieces. When we do that, we form pieces called introns and pieces that are called exons. So it's split into these pieces. We remove the introns, we get rid of those, and we use the exons. So these exons will come together to form our final piece of mRNA. We put a cap and a tail on it to kind of finish it off, and then we can use it to actually make our protein. So we remove the introns and we use the exons. Exons are what's expressed. It's kind of the way to help you remember that you use the exons, because exons is what's being expressed. Remember, a protein is a string of amino acids, and the amino acid is attached to the tRNA. The sequence of letters, the sequence of the bases, the A, the T, the C, and the G, is going to determine what amino acid is going to be used. When we look at a piece of mRNA, every 3 bases is going to signify or specify one amino acid. This is called a codon. So here's a picture of our genetic code. This is our piece of mRNA, and we have 6 spaces here. So this is our mRNA, and the first 3 here, this is a codon. The second 3 is another codon. So the first codon we have here is AUG. The second codon is AAC. There's a special chart that we can look up to see what AUG and AAC translate to be. So we need to use some sort of dictionary. When we use this dictionary, we are going from mRNA to the protein or the amino acid sequence. I like this chart. When we use this chart, we start in the middle. So the first one we had was AUG. So we're going to start here, A, and then we're going to go out to our U, and then we're going to go to our G. And we can see that it codes for MET, or methionine. You will not need to memorize this chart. This chart will always be given to you. The other one we had was AAC. So let's start in the middle. Let's kind of erase this out so we can see. So start in the middle, A, C, ASN. So that's asparagine. So those are the amino acids that are coded for by those 2 codons. So we can use this chart to determine what the order of amino acids is. Then when we get the amino acid sequence, that will become the protein. That protein then will dictate what's going to happen. If you notice here on this chart, we have some ones that say term. These are stop codons. Stop codons do not code for anything. They just say to stop. We have 64 different combinations of sequences for the 3 bases. We have 20 amino acids. So that means that there are more bases or more combinations than there are amino acids. So some of the combinations are going to code for the same amino acid. There is one start codon, so every amino acid sequence is going to start with AUG or methionine. And then there are 3 stop codons. There's no amino acid with this, it just basically says to end that protein. So this process of going from mRNA to protein or amino acid sequence is called translation. It takes place on the ribosome, so the ribosome reads the mRNA, attaches the appropriate tRNA, so that tRNA has the anticodon, basically that 3-letter sequence that's going to pair up with that codon on the mRNA, and it also is carrying that amino acid, and it's going to chain together. So here is a picture. We have our mRNA coming out of the nucleus. We have our ribosome here, so it's sitting on the ribosome. We have the tRNA, those kind of trumpet hairpin looking things. Our start codon is AUG here. The anticodon, UAC, is attached to that tRNA. That tRNA is carrying that amino acid. The next tRNA comes in, matches up, and these 2 will connect together. As we see in our next picture, that first tRNA leaves, and the next one comes in. The amino acid chain together, and this keeps happening until you get to a stop. So here in this last picture, we have our polypeptide or our chain of amino acids, and then we get to UGA. And if we went back to that codon chart, we would know that UGA is a stop. So here, the whole process stops, and then this protein can be further processed. This all happens on the ribosome, and that piece of mRNA can be used over and over again until it's not needed anymore. So our step goes that that we have DNA. So if we have a DNA strand, let's just do a real quick example. We're going to make mRNA from this. This is called transcription. So we're going to pair up our complementary strand for our mRNA. A pairs with U, T pairs with A, C pairs with G, G pairs with C, T pairs with A pairs with U. Remember, because U is in the mRNA, T is in DNA. Then we want to go to our amino acid sequence. The amino acid sequence, or going from mRNA to amino acid, is called translation. And remember, with translation, we need our codon chart. We need a dictionary to go from the mRNA to the amino acid. So we need to look up our UAG, and then we need to look up CAU. So when we look at our codon chart, we can actually come to find out that UAG is a stop codon. So we wouldn't go any further. But let's just pretend that we would keep going. CAU is going to translate to histidine. And then what we would do is we would look up, or the cell would figure out, what does this amino acid sequence do? So something that you need to be able to do is go from a piece of DNA to a piece of RNA through transcription, and then look at that piece of mRNA to go to the amino acid sequence, or translation, and then be able to look up on a chart what that protein sequence stands for.